Hi everyone, welcome back to Useful Genetics. This is lecture 2F, where we're going to talk about mutations that affect function even though they're not in coding sequences. So we'll talk about regulatory sequences in DNA that are recognized by proteins, about sequences in RNA that affect splicing and translation, and about regulatory RNAs. Now, there are a lot of different non-coding sequences that do affect phenotype. They're only a small component of the genome, but they're vitally important. And you've already learned about almost all of these. So the first class I want to remind you of is sequences that affect transcription. So sequences can affect when the DNA is transcribed, what cell types the gene is transcribed in, and how strongly the DNA is transcribed. For instance, the sequence of the promoter determines how, once transcription has been activated, how strongly RNA polymerase is going to bind there and initiate transcription. Mutations that affect the promoter will change that. There are many regulatory sequences, mostly in front of genes, and changes to any of these can affect when or the cell types or how strongly the gene is transcribed. Sequences in the DNA can also affect things that happen to the RNA. For instance, the ribosome binding site in the messenger RNA affects how strongly and efficiently the RNA is translated. And intron sequences affect splicing. We talked in a previous lecture about the fact that almost all the sequences in introns are effectively junk. As far as we know, they have no contribution to phenotype. But introns do contain some critically important sequences because the sequences at the ends of the introns provide signals to the splicing machinery telling it what sequences should be cut out. And you'll see an example of these mutations, I think, when we talk about cystic fibrosis in Module 3. Now, other genes function as RNAs in the same way that most genes get translated into functional proteins. And by far the most important of these are the transfer RNAs and the ribosomal RNAs. We already looked at transfer RNAs in the previous lecture. Here's an example where we'll think briefly about mutations in the RNA itself. We talked about how a mutation in the codon can change the meaning of the sequence. But now we want to think about mutations in the transfer RNA itself. So for instance, a mutation that changes the transfer RNA shape can change whether or not the enzyme that normally adds the amino acid onto the transfer RNA will act. They can either mean that the transfer RNA doesn't get charged with any amino acid at all. It basically goes into the ribosome, but it can't do its job, or it may get charged with the wrong amino acid. Even more dramatic are the effects of mutations that change the actual anticodon. So mutations at any of these three positions here in the transfer RNA will change which codon the transfer RNA binds to. For example, a mutation that changed that G into a U, a mutation in the DNA that changed that G into a T, would result in an aspartate transfer RNA charged with the amino acid aspartate, but with a codon that does not recognize the aspartate codon in messenger RNA. Instead, it would bind at a different codon and insert aspartate at a place where there should be a different amino acid. Now, I'm not going to talk in detail about mutations in ribosomal RNA, but I did want to show you this model illustrating the structure of the ribosome. It's easy to think of molecular machines as being mostly made of protein, but the ribosome is mainly an RNA machine. Proteins are synthesized by an RNA machine. You can't even see, it's just this intense tangle of RNA, but almost every base in that RNA has been optimized by natural selection so that the machinery folds into exactly the right structure to carry out protein synthesis. Most of the proteins that are components of the RNA are just there to help the RNA fold into the right structure. All the critical work is done by the RNA. And of course, ribosomal RNAs are highly conserved RNAs because so much of their sequence is critical to their function in protein synthesis. Now, another category of non-coding sequences that are very important for function are other molecules that function as RNAs. Some RNAs are themselves catalytic, like the ribosome, but much smaller. This is an example of a very much simpler RNA folded into a structure called a hammerhead structure. It's a catalytic RNA, and it's held together by base pairing. For example, these bases pair with each other to create the secondary structure that gives it its catalytic function. Another category of functional RNA, again, that can be changed by mutation, are what are called antisense RNAs. You'll encounter one of these in Module 3. These are RNAs whose function is to prevent the expression of genes not by preventing transcription, but by preventing translation. And they do that by being complementary to part of the messenger RNA. So this antisense RNA can form base pairs with this messenger RNA and prevent it from being translated. So here's a question, sort of a summary question. When can a mutation that's not in a coding sequence change a phenotype? And note that these are square boxes, meaning you can choose more than one answer if more than one is correct. Well, all the answers were correct. All of these are ways that mutations can change a phenotype even though they're not in a coding sequence. Now, what we've done, we've talked about the important functions that non-coding sequences can have. All of these functions can be changed and destroyed by mutations. So regulatory sequences, recognition sequences in DNA, such as transcription factor binding sites, genes for the transfer RNAs and ribosomal RNAs that function in protein synthesis, and genes for other kinds of functional and regulatory RNAs. Now, this isn't an exhaustive sequence. There's lots of other sequences that don't code for protein but have important functions in DNA, but we don't have time to talk about all of them. Now, coming up next, we're going to talk about other causes of mutation. So far, we've just thought about mutations caused by errors by DNA polymerase, but there's a lot of other ways that DNA sequences can change, and we'll talk about those in Lecture 2G. I hope to see you there. Hi everyone, welcome back to Useful Genetics. This is Lecture 2G, where we're thinking about, finally, other causes of mutations than mistakes by DNA polymerase. We'll think about insertions of mobile elements. They're a major cause of mutations, and we'll think of some of the consequences, and then we'll discuss changes to chromosomes, duplications and deletions, rearrangements, and changes in chromosome number. Now, we'll start with insertion of a mobile genetic element. And here I've shown the before and after of an insertion of a mobile element, what we call in earlier slides we referred to as genetic parasite, into a gene. Now, these mobile elements can insert anywhere in the chromosome, but here I've drawn it inserting into a gene. And to give you a sense of how this works, I've made a little animation. So what happens is that a copy of the mobile element comes to the gene and cuts it, and then inserts itself into the gene and seals itself into the gene, so that now we have a continuous DNA double-stranded molecule, part of which is the original gene, and part of which is the new sequence of the genetic parasite. Now, as I said, this can happen not only in genes. If it happens in a gene, it's likely to have very serious effects on the gene's function because it disrupts the coding sequence. Here I've shown insertions accumulating outside of genes, and One way to explain that would be to say that insertions also happen inside genes, but because they're usually destroying the gene's function, natural selection eliminates those mutations from the population. So we don't very often see insertions of mobile elements into genes, but we very often see them elsewhere in the genome. So this is evolutionary time. Initially, a few insertions, and then as time goes on, more insertions of different types of elements. As you saw, the different colors in that pie chart represented different kinds of genetic parasites. And some of these genetic parasites are inserting into sequences that themselves were insertions of genetic parasites. So it's the little fleas have little fleas to bite them. Here's another one where an insertion of a genetic element is inserted inside another one. As time goes on and the genome evolves further, more and more of these elements accumulate. Now we've got pink ones piling up, pink ones inside other ones. The genome is getting bigger and bigger. It's not getting more genes, but it's getting bigger and bigger because it's getting more and more mobile genetic parasite sequences in it. In fact, these mobile genetic, a particular class of these genetic parasites, is thought to have been the original cause of introns, that they began as, They began as insertions of a particular type of genetic parasite into coding genes. And this type of parasite, because it was able to splice itself out of the RNA, didn't destroy the gene, wasn't eliminated by natural selection. And this is how introns are thought to have begun. So I've diagrammed one here. Here's another one where the intron genetic parasite has inserted, and then another genetic parasite is inserted inside the intron. So the introns themselves are also getting bigger because they are also sites where mobile genetic elements can insert into the genome without causing any harm to the function of the gene. Now, this leads us to thinking for a minute about, well, how big are genomes? What determines how big genomes are? So this is a graph that's on the x-axis. It has the size of the genome of many different organisms, viruses, bacteria and archaea, eukaryotes, the size of their genome, and the number of protein-coding genes. Now, we talked about RNA's a function of genes, but almost all genes in organisms are protein-coding genes. And what we see is that viruses have very small genomes. Most viruses, a few viruses are very large, but most have very small genomes, but they manage to squeeze in quite a lot of genes, like up here, for instance. Bacteria and archaea have very compact genomes. There's not much junk, not much mobile elements in their genomes. And their number of genes that they contain is quite nicely, linearly a function of how big their genomes are. But the situation's very different for eukaryotes. Now we see a whole wide range of genome sizes from less than 10, almost down to 1 megabase, all the way up to 10,000 megabases, so that's 3 times as big as our genome, and very less dramatic, but still dramatic differences in number of protein-coding genes. Here's us, we're this yellow circle. We're actually pretty typical for animals. Our genomes have about 20,000 genes, maybe 30, depending on how you count, and we're about 3,000 megabases of DNA. And most of our genome is not genes. If most of our genome was genes, we'd be over here on the graph. So here's the question. Mobile elements have important consequences. As I said, usually the insertion of a mobile element will eliminate the function of the gene, and that's because if we consider this gene And for now, let's just ignore this little regulatory sequence that I've drawn here. Just consider the normal gene, the blue gene with its promoter, ribosome binding site, and start codon. Before the mobile element inserted, this gene encoded protein A. Now that the mobile element has inserted into it, it works in effect as a frameshift. This is a sequence that wasn't selected to be transcribed and translated from these sequences. And so when translation starts at this start, so it will be transcribed into a messenger RNA from its original promoter. But when translation starts from its original start codon, it's going to be translating sequences that are part of the mobile element. And chances are that the protein will be completely non-functional because these sequences were not evolved to act as proteins, and will truncate prematurely because it'll meet a stop codon. But in the case that I want you to consider, which is actually a fairly common case because many mobile elements contain regulatory signals themselves, I want you to consider the case where the mobile element itself carries a second set of regulatory signals. There's a promoter pointing out of the element, and there's a ribosome binding site and a start codon. And then that's followed by the rest of the blue gene, gene A. And what this question is asking you is, will the protein that's produced from this promoter, from this messenger RNA, will it resemble the normal protein A? And the answer is, well, it could, but it probably won't. Even though in this drawing, almost all of the gene for protein A is intact, and it's got a substitute promoter and binding site and start codon. But there's one problem, which is, what's the probability that we have the correct reading frame here? 2/3 of the time, by chance, 2/3 of the time, we're going to have the wrong reading frame. And so we're going to produce a protein that does not resemble protein A. The protein will be translated, but again, because it's the wrong reading frame, it's likely to hit a stop codon and the protein will be truncated and non-functional in any case because it's making the wrong amino acids. 1/3 of the time, though, it'll have the right reading frame, will produce what could be a pretty much functional copy of gene A, of protein A, because only a little bit's been removed from the end. And the consequences of this kind of event can then be a change in how gene A is regulated. Instead of being regulated by these signals, now it's regulated by these signals. And that can have very interesting and important evolutionary consequences. And we see these quite a lot when we look closely at the evidence of how gene regulation has evolved. Now, let's switch to thinking about chromosome mutations. And we'll just cover these very briefly because we'll do them in a lot of detail in Module 10. The first kind to think about is duplications or deletions of large segments. We talked about very short duplications and deletions, insertion of a base or two, deletion of a few bases. But larger deletions can have quite dramatic effects. So here's a duplication where this segment shown in red is now present twice. We call it a tandem duplication because the two copies are side by side. So we've still got a copy of the green gene and the pink gene, and we've got a new gene, a hybrid gene, whose first half is of the pink protein and the second half is the green protein. If the reading frames are compatible, this may produce a completely new protein. Here's a drawing of a deletion where the same segment, instead of being duplicated, it's been deleted. And again, we've got a hybrid protein, but at the cost this time of losing our functional copies of both the green and the pink proteins. We can have larger breaks and joins, not within a single chromosome, but between different chromosomes, so that chromosomes can become sort of hybrid chromosomes with parts from different sources. And over long evolutionary periods, we see that quite a bit of this goes on in typical genomes, so that rearrangements of the parts of chromosomes are quite common. Now, finally, we can see changes in chromosome number. For instance, we might lose a whole chromosome due to a mistake in cell division, or in a complementary event, wind up with 3 copies of a chromosome. These events are important both because they change the dosage of genes. So a cell with this genotype would only make half as much of this protein as it normally would, and it would make 50% more of proteins on this chromosome than it normally would. They're also important because they create problems for sexual reproduction. And this will be discussed initially in Module 7 when we talk about sexual reproduction, about meiosis, and then again in more detail in Module 10. So we've talked about 2 kinds of other ways that genomes can change apart from errors by DNA polymerase. We talked about insertions of mobile elements, which are a major factor in mutational change and in genome evolution. We talked about how they happen. They actively insert themselves into other DNA sequences. We talked about the consequences for gene function and for gene regulation, usually destroying gene function, but sometimes creating new kinds of gene regulation, and the consequences for genome evolution. They're a large part of the reason that our genomes are so big and so full of sequences that aren't genes. And then we talked about chromosomal mutations, about deletions or duplications of segments of chromosomes, breaks and joins that rearrange segments of chromosomes into new combinations, and about changes in chromosome number. Coming up next, we're going to talk about a distinction between 2 kinds of cells that mutations can happen in, germline cells and somatic cells. And this will then prepare us to think about things like the roles of mutations in the development of cancer. I hope to see you there.